Hello there, and welcome to my pathway map. My name is Luke McKeon. I'm a senior at CSS, and this is my pathway map on the SSRI, or selective serotonin reuptake inhibitor, fluoxetine. Also, you may know as Prozac. So, on the left here, we have the legend where I laid out essentially the four main components of both my top and bottom pathway maps, top being the normal or natural transmission of serotonin across a synapse, and the bottom being the same thing but in the presence of an SSRI such as fluoxetine. And I chose fluoxetine mainly because it's pretty common as far as drugs prescribed for mental illnesses. It's used to treat depression, obsessive compulsive disorder, and quite a few other mental illnesses. But also, it's in this class of SSRIs, which there are tons of other drugs in this class, and they all sort of generally exhibit the same action within the synapses. But I chose fluoxetine because it's pretty common, and it was an easy one to find information on how this whole mechanism works. So essentially here in my diagrams on the legend, I have blue molecules being serotonin, which are your natural neurotransmitter. The fluoxetine or drug shown in red, which is only shown in the bottom pathway map. You have your green serotonin reuptake transport proteins in the green triangles, which are actually going to be the site of action for the drug fluoxetine, as you can see in the bottom pathway map. I'll talk about that in a second. And then you have your normal, mostly on the postsynaptic neuron serotonin receptor proteins, which essentially are there to receive the synaptic signal of serotonin. So as the serotonin moves across synapse, they're the ones who recognize that and carry on the neural transmission signal. So to see all this in motion, let's look at the normal neural transmission pathway. So first of all, I should say there's an asterisk on top of both of these rectangular boxes indicating that this whole process is being initiated by a presynaptic action potential. And that's going to trigger the synaptic vesicle to exocytize this serotonin into the cleft, the synaptic cleft here. And so for the most part, these serotonin molecules are going to hit the receptor proteins. So those are mainly on the postsynaptic neuron, but there's also some on the presynaptic neuron to provide some feedback inhibition for this whole process, so your body's not exocytizing too much serotonin. And then also there are the green SERT proteins that are actively recycling that serotonin back into the presynaptic cell so it can reuse those for further signaling later along the cell's life. Probably pretty soon. But anyways, in contrast on the bottom, you can see what happens when we introduce SSRI into the mix, such as fluoxetine. Essentially, the same process is trying to be undertaken, but you see the red drug cells are binding and inhibiting these SERT green triangular proteins on the presynaptic neuron. I'm sorry. So the presynaptic cell can't reuptake the synaptic serotonin. So what you end up with is a heightened synaptic concentration of serotonin molecules, or effectually an increased neural signal. And so for people with mental illness who normally might have weakened serotonin signaling in their brain, these drugs can actually aid them in achieving more normal neurotransmission levels. So in that way, these drugs can be very helpful. But along the way of learning about this whole process, I kind of got intrigued by the question of how do these drugs pass the blood-brain barrier, because I know the blood-brain barrier can be such a canonically difficult obstacle for therapeutics to pass. And so, yeah, I mainly wrote about different proteins in those types of cells of the blood-brain barrier that could affect the passability of certain drug types. But yeah, that's my pathway map, and thank you for watching.